Hi everyone, my name is Xu Shi. I'm a first-year PhD student from Dr. Romerte's lab at the University of Nebraska-Lincoln. Today I'm going to share results from my project aimed at understanding the relationship between the gut microbiota and experimental autoimmune myocarditis. Myocarditis is an inflammatory disease of the heart muscle and has caused millions of people die worldwide. Myocarditis is caused by injury to heart and can develop into heart disease and ultimately heart failure. Heart-specific autoimmune responses are a hallmark of myocarditis. In a laboratory, we can model these autoimmune responses in a mouse model of EAM. Using this mouse model, recent research has suggested that the host immune responses to the gut microbiota can influence EAM disease outcomes. However, a causative link between the gut microbiota and myocarditis is still missing. Therefore, we set out to answer the research question, can the gut microbiota influence EM severity? And we hypothesized that the gut microbiota influences EM severity via alteration of autoimmune responses. To test our hypothesis, we selected three distinct mice microbiome, M31B, W116, and MC608, to colonize germ-free C3H mice. After 2 weeks, on day 0 and day 7, EAM was induced by injecting myosin plus adjuvant to half of the mice. The other half of the mice received adjuvant alone as control treatment. Then 2 weeks after day 7 treatment, all mice were necropsied and samples were obtained to assess heart pathology and analyze autoimmune responses. Tissue sections from mouse hearts were evaluated microscopically by a pathologist who calculated the heart inflammation index based on the percentage of myocardium involved and the number of inflammatory foci. The higher the index, the more severity of EAM. Injection of myosin plus adjuvant induced the signs of EAM in all mice, regardless of microbiome, when compared to adjuvant treatment. However, mice harboring the W116 or MC608 showed significantly higher inflammation scores versus those with M31B microbiome. In these representative H&E stained photomicrographs of the heart tissues, you can see increased inflammatory cells infiltrates in the hearts of all mice with EM, but there were multifocal inflammatory lesions in the mice harboring MC608. We next analyzed autoimmune responses. To assess B-cell response, serum anti-myosin IgG were detected by ELISA. This graph shows curves of optical density values of the log dilution of samples. The presence of MC608 microbiome induces stronger antibody response compared to the other two microbiomes. And then we also isolated splenocytes from mice and stimulated them with myosin protein to detect T-cell interferon gamma production. T-cells from mice colonized with MC608 also produced significantly greater levels of interferon gamma versus the other two microbiomes. Together, these results indicated that the mice harboring the MC608 microbiome generated the strongest autoimmune responses, which correlates with more severe EAM. We next analyzed the composition of gut microbiota present in fecal samples before and after EAM induction. The induction of EM did not lead to major global changes in beta diversity. However, we did observe changes following the induction of EM at genus levels. In particular, the abundance of lactobacillus, shown here in orange, was decreased post-EM across all microbiomes, with the greatest decrease in mice harboring MC608. Closer examination of the two most abundant lactobacillus, Johnsonia and Marinus, revealed that EAM induction significantly decreased Johnsonia levels in mice harboring W116 or MC608, and only mice harboring MC608 showed significant decreases in Marinus. Altogether, this result suggests that lactobacillus species may play a protective role in EAM. To conclude, we saw that the variation in gut microbiota composition modulates the severity of EAM and associated autoimmune responses. The mice colonized with MC608 produced remarkable higher anti-myosin interferon gamma and IgG responses. The abundance of the Lactobacillus genus showed the greatest decrease in mice that experienced the most severe EAM. In the future, a causative role of Lactobacillus species in regulating the pathophysiology of myocarditis will be investigated. I want to say thank you to all the members in our lab and our math team. I also want to give special thank you to Dr. Paul Linder, who has provided important effort to initiate this project. I'm willing to take any questions. Thank you, guys.